Hello, Untitled everyone. Welcome to class 11 biology. In our previous video on chapter cell cycle and cell division, we studied about cell cycle, the phases of cell cycle, and we also discussed in detail about mitosis, or one of the cell division called mitosis. Tangwazu dinge video nalo ngazu khar kaltar lawa imba cell cycle lana khar lagyume ani cell cycle nalo la phases niwa interphase na ani M phase. So we discussed about cell cycle, right? Cell cycle lana khalla gorla so ro cell cycle la zambala the events, right, which happens during cell division. Cell chigne cell ni la divide chebe karola sequence of events chunguyo. This sequence of events the chazan chilo ngazu khalla gore cell cycle la gore la ba ani cell cycle nanglo la cell cycle ki definition chana zu tiya warda cell cycle nanglo la phases khamanda ni yores la there are two phases, right? We have got interphase and we have got M phase. These are the two phases of cell cycle. Cell cycle ki phase manda ni dire cell cycle nanglo la there are two phases, interphase and M phase. Now if we look at these two phases, and if we look at interphase, it is also called as the resting phase or the preparatory phase. Interphase la resting phase ke lagore hamena preparatory phase ke lagore. It is the most active phase of the cell division, during which all kinds of duplications happen. Cell gi nanglo lyo genome ne chen cell organelle ne chen en ko nanglo lyo gi biomolecule dige sangma double chaya cell korang grow chaya dige sangma khadu chungore interphase ka chungor. So interphase nanglo la khajore there are three phases, G1, S and G2 phases, right? Interphase nanglo la phase ta tezemara khajore khamana sumyor. It happens in this particular sequence, G1, S and G2 phase. And M phase nanglo la M phase nanglo la ngazo khare we have got two, right? Khare karyokinesis, it is the phase of actual division, right? M phase la dikhare it is the phase of actual division. Interphase dikhare preparatory phase cell it prepares itself for cell division, whereas M phase kabla kharchi ho raha the cell actually divides. And during M phase two events happen, karyokinesis, which is called as nuclear division, and it is followed by cytokinesis. It is followed by cytokinesis. Okay, cytokinesis la mena division of cytoplasm, karyokinesis la mena division of nucleus. So karyokinesis and cytokinesis la and karyokinesis na lo yang khare G2 gi jela ro p into stinda phase here ro p mat lantir. So P stands for prophase. Okay, so karyokinesis ki step thangbo dikhare prophase, right? And this is followed by M la khare metaphase and A la na anaphase, and finally we have telophase. This ends the division of nucleus. The cell nanglo nucleus the divide chesa ve chela. After the nucleus divides, what happens? Right, it is followed by cytokinesis or cytoplasmic division. And in cytoplasmic division na kiran zo khare themba nya ala so different plant versus animal cells. Cytokinesis na plant versus animal cells na la cytokinesis ni de manda yorela so odi kiran themba nya ala so ti khare da ngonzu gi nyama video na lo la turti to sta we mena khare jang yemi. This is the summary of what we have studied in our previous video. Okay, now in this video we are going to see the differences between two different types of cell division. Cell division kamanda ni ki kala kewa de ngazu jang try chi ba. Okay, so overall difference we will try to see the overall differences between the two different types of cell division. Then two different types of cell division khare la so I told you we have got mitosis, which we have studied already. Mitosis study chesarta. The next one study chigo already meiosis. So these are the two different types of cell divisions, right? The video di nalola mitosis the meiosis niki general kewa di hakwa try chenda. Before we move into the details of meiosis kabla khar khar chungwe me di majang nyala ro mitosis the meiosis ni para yi and overall kewa di khar yume tayenda. Ani kewa jelan khari gali ser dur kana meiosis detail na jangdu zambala ro and kunyi ni para difference shumarwe it becomes much clearer later on. Okay, ta desia ro ta ni thangbo mitosis ta wi bai na ro mitosis kabla I will show you diagrammatically. Okay, mitosis ki kabla we have one cell, right, one cell with the nucleus over here, right. So one cell, it divides into two daughter cells. Okay, one cell, it divides into two daughter cells over here. Okay, so this one cell, it divides into two daughter cells. This one cell is called as the, we can call it as the mother cell. Okay, so consular daughter cell lagi we will just call it as mother cell. And now you have to know that this mother cell, right, which cell undergoes mitosis? All our body cells or somatic cells, they undergo mitotic cell division or mitosis. Mitosis la di mother cell khadire khandi gi mitosis to go la wi na somatic cells ni chi wor. The somatic cells, they undergo mitosis. Now mitosis na lo la somatic cell cho mother cell gi keju ni wa dikhare la wi na, they are diploid cells most of the time. They are diploid cells most of the time. Most of the time, diploid cells undergo mitosis, not the haploid cells. Now, what do we mean by diploid cells? Diploid cells are those which contains two sets of chromosomes, right? Diploid number of chromosomes. I told you, in human beings, we have got 23 pairs of chromosomes. 23 comes from our father, 23 comes from our mother. So we have got 23 pairs of chromosomes in all our cells. 저기 셀 상만 나로라 가져요래 크로모솜 1024면 편동는 17주제 요약하려래 1024면 나오고 김생 가래 차직 수산을 리고래 1024씩 수산 리고는 빨아산 리고 1024씩 수산 리고라 보면 아무나산 리고 군이 인념도 야 루체는 가져서 1024씩 이 차서 so 2n 디카래 가져가지디 23 n 디카래 23제 total 가져래 46 chromosome chawor. So diploid number dikhazu chawor 46 chawor. Wa seo jongso. So cell dende do dige sangma wa diploid cell dige sangma they will undergo mitosis. Somatic cells which are diploid cells, they will undergo mitosis. And now after it undergoes cell division, the cell division do kabla. When it undergoes cell division, what are the phases? Apply phase thangsho dikhare lawena, it undergoes interphase, which is the longest phase, which is also called as the preparatory phase. Thati yo cell di adi nanglo le yogi genome sama double cha cell gi nanglo le yogi khare cell organelle da dike sama double cha enzymes ta dike sama biomolecule dike sama thewa synthesize cheni ani haji gore thati yo cell division chela ti khare interphase. Interphase ki je la khare M phase 2. And M phase nalola karyokinesis ta cytokinesis. Karyokinesis kabla kharchungure prophase, metaphase, anaphase and telophase. P match da. And the last it undergoes cytokinesis. It undergoes cytokinesis and it gives rise to two daughter cells. The dingi khare, they are daughter cells. They are daughter cells. Now if we talk about the features of daughter cells, daughter cell produce these two daughter cells, they are both diploid. They are both diploid. Diploid lo tama nyata tishe jasa so. Kare kono lo chromosome thanga jomare. Koigi mother cell ke nanglo chromosome thanga da chikpa chikyan charo. Di nanglo la ngazi di kesi human cell imba chasha. Di nanglo chromosome shipshedu yo imba na daughter cell niga nala chromosome khazo yore shipshedu yor. So this is the products of the mitosis. Mitosis produces two daughter cells, and these two daughter cells, they are both diploid, just like their mother cell, right? And another one, or you can just simply call it as they are identical to each other, right? Yamena they are both same as their mother cell, or they are identical to their mother cell. They are identical to their mother cell. They are identical to each other and they are identical to their mother cell. So ta dinalo keju sang seo chungso mitosis kabla khar chunggu me dige sang hyang clear chungso. Ta di mitosis ki kabla ta masu di sang jansa. Wa ta ngazu meiosis kan ta enda. Wa ta that will see the difference between that we will see, show the difference between the mitosis and meiosis. Ta meiosis kabla khar yore chikpa chigya ta dina shinta. We have got a mother cell. We have got a mother cell, right? And it divides. It divides into two cells over here. It divides into two cells like this. But again, these two daughter cells, they will undergo again. Each will undergo cell division to result in 4 daughter cells. Each will undergo cell division to result in 4 daughter cells over here, right? So the general difference thangpo derda. Mitosis, it happens in a single step. Cell cycle chik nang lo la sharja gora kharchik do. It gives rise to 2 daughter cells in one single step. Whereas meiosis, it occurs through 2 steps. Step ni kang chungur. Seo chungso. Ngosi do beto. Tela khardo. Cell chik ni cell ni chak do. Phagel khajik do. Cell chik ne cell shi chak. Cell shi chadan kare thangbo cell chik ne ni la ma cha. Dene cell ni di khajir khorani ni ni la chadu zambala ta khajir cell chik ne cell shi la ma cha so. Di chazan chik la ngazu ki meiosis chik la gorda. Dene di bodu chaya de la ngazu khala gore mitosis chik la gora. Dene di bodu doya de la meiosis chik lar. Seo chungso. Tata ta khewa ta wi bai na. Ta thangbo ngazu mother cell ki ngala khewa she enda. Mother cell ki ngala khewa she bai na. Di nanglo mother cell dige sama khare ngazu gi body cells. Somatic cells dige, they will undergo mitosis la so. Phagela meiosis la di cell khandigi under meiosis undergo chigre la na. The germ cells or the sex cells, right here, the special cells called germ cells or the sex cells, they will undergo meiosis. The germ cells or the sex cells of our body, they will undergo meiosis. For example, the cells lining our seminiferous tubules in our testes and the cells within the ovary of the female, they undergo meiosis. In sexually reproducing organism, how are gametes produced? Sperm the egg line is produced through the process of meiosis. Only specialized cells, those germ cells, right, the sperm mother cell and the egg mother cell, they will undergo meiosis. Other body cells, they won't undergo meiosis. Only the germ cells will undergo meiosis. The mother cell is diploid in nature. Mother cell dikhare diploid re. Okay, so here if we compare the starting point, the mother cell para lekhewa kharchas chikhare somatic cells re, the body cells re. Mother cell of meiosis is germ cells, special cells called germ cells, which gives rise to gametes later on, sperms and eggs producing in all water. Okay, whereas somatic cells, they will just give rise to other somatic cells. Okay, ta mother cell gi tsawe jela ta khachido mother cell di it undergoes cell division, right, and gives rise to 2 daughter cells over here. Daughter cell dela kha manda ni chagdo, it produces 2 daughter cells over here. Now ta de para de khewa de ta we na din nanglo la chikbo jigya yorda interface yore ro interface yore tina m phase kabla p mat 2 gore ro, and then cytokinesis chungor chikbo jigya de yungorda ro. Interface, prophase, metaphase, anaphase, telophase, cytokinesis, it gives rise to 2 daughter cells. But meiosis doesn't end over here. The two cells which is produced by this first step, it continues to divide in the second step. Second step now divides here, and this gives rise to 4 daughter cells. These 2 daughter cells, they are haploid. Haploid in nature. Ta dela kharjere chromosome number thanga jomarwe chega char. the chromosome number is now half the number of chromosomes as compared to their mother cell. These two daughter cells, their chromosome number, it is half that of their mother cell. Ta kisi the human cell da uji bai na. Mother cell nanglo la kharire chanyi saksum yor 17 yor chromosome 17. Di dingi ni nanglo la chromosome khasu yor la bai na only 23 ma de yomar. Seo chung so step thangbo di tirda. And then these two daughter cells now which are haploid. These two haploid daughter cells, they will undergo again another division, right? Interkinesis interface. After a brief interkinesis, interkinesis la wing by na interface chasan roji mai bache. Interface la wing na step mabuya G1, G S phase and G2 phase na niwa. Did the jigo jige mai bache de la kharere resting phase tundujor tilangazu khalere interkinesis. The cell, it stays in interkinesis for a short duration, and after that immediately it starts dividing. Din disabala kharere prophase, metaphase, anaphase, telophase lanier. And this is followed by again cytokinesis. And 4 daughter cells produce chigdo. So these 4 daughter cells, they are all haploid in nature. They are all haploid. Human cell they all contain haploid number of chromosomes. They are all haploid in nature. So it results in the formation of 4 daughter cells. Okay, clear chung so. Ta ti ma jel runner da. Ta khewata wa imbai na khonsama khare. It results in the formation of the daughter cells. The final daughter cells the final daughter cells. These are the daughter cells, and final daughter cells, they are all haploid, and they are one important difference. All difference, they are all unique. Unique what do we mean by unique? Right? Unique individual unique. So these all daughter cells, they are unique. They do not resemble their mother cell. Genetic composition we are talking about its genome, right? The genetic composition, they are unlike their mother cell. Chromosome chromosome but unique all these cells, they are unique. They are all different from each other. They are different from their mother cells, obviously, but also they are different from each other as well, right? The genetic composition of these cells, they are different from each other. So they are all unique, unlike these two cells. These two daughter cells of mitosis, they are identical to each other, and they are identical to their mother cell. But over here, the 4 daughter cells produced over here, they are not identical to each other. Obviously, they contain half the number of chromosomes. But even all these daughter cells, even when all these daughter cells, they contain haploid number of chromosomes, they are genetically unique from each other. The condense unique meiosis detail clear. So these are the differences now. Step we have got 2 steps over here. Step thangbu nalu khachido cell chik di ma chani cell ni chagdo. Step niwa nalu cell ni di khachido ni la ma chani cell shi chagdo. Step thangbu di langazu khalle la wembana, it is called as meiosis 1. Di langazu khalle meiosis 1 lir. And the niwa di langazu khalle obviously it is called as meiosis 2. Tadi ngelo iksu pe kyanzo chadu de kyanzo the chair try to understand. Okay, we have got a small board over here. Okay, so meiosis 1 and meiosis 2 lag. Interface interface 1 then p met prophase 1, metaphase 1, anaphase 1, telophase 1 and 2 when we study in detail, when we study meiosis in detail, we will see that the prophase, metaphase, anaphase and telophase, they are all prophase 1, metaphase 1, anaphase 1 and telophase 1. Why? Because these are the phases of meiosis 1. Any the newer the prophase 2, metaphase 2, anaphase 2 and telophase 2 nan changar. 다 meiosis 1 다 meiosis 2 니 바라라 개와 하르되 다 mitosis meiosis 간 개와 디시게서 딩이 바라라 개와다다 meiosis 1 is reductional division. Meiosis 1 is reductional division. 칼라오레 meiosis 1 the reductional division 카레라고 라고면 가진다고 라고면 됐습니다. 크로모솜 기 장가지면 돼. 크로모솜 기 장가디 diploid ne haploid la khawala chagdo step thangbu chagdo meiosis 1 gi kablo chungdo. So therefore meiosis 1 is called as reductional division. Meiosis 2 gi kabla haploid nangja degido. So meiosis 2 di kharla gore equational division da detisamindo detishinda. It is called as equational division. It is called as equational division. Meiosis 1 is called as reductional division because the chromosome number it is getting reduced from diploid to haploid, whereas mitosis 2 it is called as equational division, just like mitosis. Meiosis 1 dikhare meiosis 1 the reductional division re meiosis 2 dikhare mitosis the chibujigan chadu so it is called as equational division, where the chromosome number is maintained and ingyodi and rangja nyadi do. Then finally khachire tenzu chumare all these cells they will become future gametes. They will turn into gametes. Kisi pod original yobi mana dige shi dikhar chadu re sperms chadu. Kisi mod original yobi na dige shi na woni chi khar chadu re egg chadu. Eggs and sperms. Okay, so these will turn into gametes, right? Meiosis results in the formation of 4 daughter cells which will in turn become gametes. And gametes kharchire sexual reproduction nalogi yona menando chatu wora. Okay, so these are the differences between mitosis and meiosis. I hope you are clear regarding the general difference between mitosis and meiosis. Okay, so in the next video we will discuss meiosis in detail. Okay, detail kan jangar the meiosis kan. Did kabla ngazo kharte gre meiosis 1 kabla khar kari chungu yeme meiosis 2 kabla khar kari chungu yeme meiosis 1 gi kabla prophase metaphase anaphase telophase chumar dikit sama ka nanglo khar kari chungu yeme detail na jangar ta. Okay, so please read about meiosis in detail before you watch the next video. Okay, thank you.